Okay, hello and welcome to this December 2017 Getting Started with CyVerse webinar. I'm Jason Williams, coming to you from Cold Spring Harbor Laboratory, New York. Welcome. This is our monthly webinar for new account holders, where we give you a hands-on opportunity to learn a little bit more about your CyVerse account and the tools, services, and other resources that CyVerse offers you. Over the next hour or so, we'll give a quick introduction to CyVerse as a project, and then give you a few hands-on demos that will orient you into the CyVerse offerings, the stack of platforms, tools, and services that can help you do research, that can be used in your education and for various other purposes. Before we get started with the presentation, let me make you familiar with the Adobe Connect technology that we're using to broadcast the webinar. Importantly, at the bottom of your screen, you have an opportunity to actually ask questions. And so one of the reasons why we do this webinar live on a monthly basis is so that you can interrupt me anytime with questions that you have about anything that I'm going to show, or questions regarding anything pertaining to your CyVerse account. Also, on the side of the screen there are some links that you can use, including downloads of presentations, and also some links to important places on CyVerse, such as the CyVerse Learning Center. That will give you an opportunity to either follow along with the demos loosely, or actually just come back at your own pace and explore some of the learning resources. At the top of the screen, there's actually a little button with 4 arrows, and you have the opportunity there to make the video full screen. And that'll be important during the time when I'm actually giving demos from my computer. I can only make my size, my screen a certain size, but you can actually enlarge the size of your own screen. Additionally, if you move your mouse over the video, you'll be presented with the option of adjusting the video quality. So depending upon your internet connection, you can increase or you can lower the resolution, whatever is necessary. Off screen to the side, I can personally see any questions that you ask. So again, feel free throughout the entire presentation to ask questions. And also, this presentation will be posted, in this case probably next week, to YouTube, so that if you've missed anything and you want to go back, you'll have an option to go ahead and watch the recording of the video. So let's get started. First, I'm going to just give you a brief introduction to CyVerse as a project. We saw from the opening survey, the majority of our users learn about CyVerse from word of mouth. Somebody in the lab may have told them that they're using CyVerse and suggested as an option. So we do want to give you a sort of background, and then we'll go straight into the demos. Mandate of serving the entire life science community. Another important thing about CyVerse is that we're funded entirely by the National Science Foundation. So we're your colleagues as well as your collaborators. That means it's a $100 million investment that's freely available to the community. And we also are interested in spurring national, international collaboration. So it's open to all users regardless of their location. We do ask, though, that you think about citing CyVerse when you use our resources, and you can find out more about citations on our website. So I've used the term cyber infrastructure a few times, and what is cyber infrastructure? Well, broken down, cyber infrastructure is really data storage, software, high-performance computing, and even people but it's organizing those systems and those capabilities to help you solve problems of size and scope that otherwise you wouldn't be able to solve. So what we attempt to give you throughout this cyber infrastructure is access to platforms, tools, and data sets, access to storage and compute, and training and support that you need in order to use those effectively. One take-home message for all of our users is that CyVerse is built for data. So whether you're working with microbes, plants, animals, biomedical data, or ecological data, whether you have sequence data, working with images, other data types, really you'll find that there are tools and resources that are going to help you get the job done. This diagram gives you a high-level overview of the products that CyVerse makes available, and some of these are what we will be covering in our demo today. So on the top layer there are the products. These are the tools that you're actually, as the average biologist, will come about to use, including our cloud computing system, Atmosphere, our web-based portal, Discovery Environment, which is a platform for analyzing bioinformatics data. DNA Subway allows you to analyze those data in the classroom context, in the educational context. We also have BISC, which is useful for image analysis and management. And we also have a variety of tools and services that are powered by CyVerse. Beneath there are other services and actual hardware, and depending on your level of engagement, if you're a developer, a bioinformatician, you may be interested in customizing how you access those things, but underneath are essentially the tools that keep your data secure through authorization, that allow you to store those data on the data store or search for it using Elasticsearch. And as you go lower, we actually reach the actual HPC systems, which means that if you're just accessing CyVerse from your laptop, no problem, because it doesn't depend on your laptop. We actually provide all of the hardware that you need to do a pretty high sophisticated analyses. Looking at some of the things that were priorities for the community, that reflects the tool set that we have. And so if you're coming to do any type of genomic analysis, you'll find tools for assembly, for RNA-seq, for variant calling, and a whole bunch of other technologies related to genomics. But it's not limited to that, but surely genomics is the driver of a lot of the big data analyses that most labs are engaging in. Also, it's important that we recognize, and as we sort of wrap up this quick introduction, the various institutions that make up CyVerse. So we are a virtual organization. We don't have a single building or place where we reside. So the majority of folks are at the University of Arizona, U of A in Arizona, as well as the Texas Advanced Computing Center down in Austin. I and several others here are at Cold Spring Harbor Laboratory, and we also have collaborators at UNC Wilmington. There is also international collaboration, and a stellar example of that is CyVerse UK, which is also a separately funded group based on these universities and institutions. Okay. So that is actually a bit of a background to the project. Now I want to just drill down a bit on the actual individual bits and pieces that make up CyVerse and that you have access to with your CyVerse account. Again, I'm just going to give a quick overview of those bits and pieces, but as I go into the demos or at any time, just want to remind you that you can always ask questions. Okay, so back to the slides. What do we think about our CyVerse products? Well, we tend to think of them almost as Lego blocks. They're individual pieces that we try to make play well together. And you are, really the author of your workflow. So we have built all the pieces. We have end-to-end workflows, but you may want something custom. We really give you the freedom to mix and match according to your research needs. That's one of the underlying pieces, one of the bigger pieces that you need to think about is data storage. So the CyVerse Data Store gives you access to really all the resources you need to share and manage data with your lab, with colleagues, and the community. Every user has a 100 gigabyte allocation. If you need terabytes, that's available too with some justification. The data are automatically backed up, and we make it fairly easy to upload, down, and share. The Discovery Environment, which is another element we'll cover in our demo today, is a platform, and you can run almost any bioinformatics application that you would normally run at the command line, except you can run it through an easy-to-use graphical interface. It's integrated with our HPC systems, and it's also extensible, which means that you can add your own applications or ask us for help in adding some more application if you don't see that it's already there. Atmosphere is our cloud computing resource, and it really gives you access to hundreds of virtual machines, fully set up fully customized computer setups. If you've never used cloud computing in research or want to get a little more understanding of how that might be useful to you, we'll cover that in this demo. That's the last one we'll do. The other pieces we won't necessarily demo today, but you should know about them. The science APIs essentially allow you to customize how you use CyVerse resources, and they're useful if you're a bioinformatician developer. DNA Subway is a really great tool for the classroom, and again, it takes some commonly used bioinformatics tools and puts them in streamlined workflows with a view towards providing course-based research experiences for students. BISC is for working with image analysis, metadata, and management of your images. It handles several hundred different image formats from microscopic to macroscopic. And again, there's a lot of flexibility and customization possible. Okay, so in this first demo, I'm going to talk about getting your data into CyVerse. And again, this might be a point where you want to click that little button with 4 arrows at the top of your video screen to make the actual video full screen, and you'll see a little bit better the demo. This first demo, again, is on the CyVerse data store, and just as a recap, you can store any type of file related to research. No matter what CyVerse platform you're using, you have access to the data store, and so it's easy to make your data accessible from one platform to the next. There are ways to automate with the data store, or to work interactively with it in that sense, and also to share, and I'll cover some of those main features. There are several ways to access the data store. I'm going to cover these three. CyberDuck allows you, through a third-party software tool, to access the data store in a drag-and-drop format. You also can use the discovery environment, which is fantastic for sharing. And if you are comfortable with the command line, which we encourage everyone to be, you can access the data store and perform transactions using the iCommands tool, another separate software package developed by iRods, which is the technology that we use to manage and serve our data store. The discovery environment, just going through the advantages and disadvantages, is really the best platform for sharing, although because it's a web-based platform, there are some size and speed limitations, so you may not want to transfer really large files. CyberDuck is a really all-purpose tool for most people. They just have a couple gigs, maybe a couple hundred gigs sitting on their computer, and they want to move it to CyVerse. This is a really drag-and-drop way. It makes it almost like Dropbox. And command line iCommands, you can certainly create your own scripts, and you can move data from server to server without doing sort of all this unnecessary uploading and downloading to third-party intermediaries. Okay, so first let me give you a little bit of a demo, and also let me take you to the CyVerse Learning Center. So if you look, pardon me, here's the CyVerse website. But in our presentation on important links in the Adobe Connect room, you'll see a link there to the data store guide. I'm going to go to that directly, pardon me, and I'm just going to scroll down to the data store guide. And essentially everything that I'm going to discuss over the next, or over all of these demos, they'll be in the appropriate guide, so you can come back here as you are following along or as you start using this for the first time. Okay, so the first thing I'm going to do is talk about this section, which is moving data along with CyberDuck. You would want to go through these instructions and download the CyberDuck tool, which is available for Windows and Mac. It should take you less than 5 minutes to download it and also do a one-time configuration. Now, I have CyberDuck installed on my computer. So when I start CyberDuck, in fact, it'll look a little bit like this. Let me open a new browser. The very first time you start CyberDuck, once you've configured, it'll look like this screen on my right, where you'll have a permanent link to the CyVerse data store. You'll double-click that, and within a few seconds, you'll be connected. And when you are connected, it'll look like the screen on my left. In this case, I see all of the files and folders that are stored on the CyVerse data store in my own personal account. So if I want to move files, for example, if I want to do a download, I could click on a particular file and drag it to where I want to go. In this case, I'll drag it to my desktop. A dialog will open, a pop-up box, asking me if I want to go ahead with that download. I'll go ahead and say clean up these old ones that I don't need. And down towards the bottom, I'll see the status of a particular download. Now, the very first moment that you're doing a download, it's really just setting up the transaction. But what you can expect on an ideal connection is somewhere around uploads and downloads of 100 gigabytes in about 30 minutes. Usually download can be a little bit slower, but you can move a fair amount of data in a reasonable amount of time using this drag and drop feature. The reverse is also true. If I create a new folder on my desktop, imagine that it was filled with data. I'm just going to call this demo 12, 13, 17. And if I wanted to move that onto my CyVerse data store, the same thing, I could move it over to the left. I would also be asked, do I want to go ahead and do that upload? Oops, this one is done. Just got to wait for it to recognize that. And in the dialog again, I'm just going to not clean up this time, I would see the status of an upload. There are certainly a number of other features that I'm not covering here, so how to actually pause or restart one of these uploads or downloads. I could also preview some of the information on some of the files that are on the left here. I could rename and other features that you just sort of expect that you would get with this type of system. But really, there's nothing more to show you as far as CyVerse CyberDuck is concerned. It really is as simple as drag and drop, and then you're already on your way to moving really large data sets. Now, the next thing I want to show you before I get into how you share your data and use the discovery environment is how you do this at the command line. It won't take long in case you're not a everyday command line user, but I will mention that in the next part of the guide, it goes into transferring using iCommands. Again, all of the instructions are here, and if you are comfortable with the command line, these will probably make sense to you. They mirror very similarly the tools or the commands you'd use for working with Linux, except in most cases they have a little I in front of them. Now you'd have to do, again, a download of iCommands, which is a third-party software, and you would have to just do that setup. But again, under 5 minutes typically would be enough to get that started. I've already got iCommands installed on my computer, so I'm going to actually open up the terminal. And let me just choose a color that's easier to see. Okay. And since I have iCommands already installed, if I want to list the contents of my CyVerse data store, instead of ls, which is the normal Linux command, ILS is the I command, and that will list the contents of my CyVerse data store. Okay. And if I wanted to go ahead and download, let's say a folder, I could do an iget command, which is the command for downloading. Since it's a folder, I use the recursive flag. I want to see the progress, and I want it to be verbose. I can download this VCF folder, and I can download it to my desktop on my local computer. Okay, if I've typed that all right, on my desktop on my local computer, I already see the VCFs folder. And on the left-hand side, I see the progress of this now completed transfer. And of course, if I click on VCS, then there is what is downloaded. If you are familiar with the command line, then it's just a few commands, many of which are listed in the help here. And there's an additional link to the full iCommands documentation. But then you are well on your way to doing automated file transfers where you may want to set up synchronization of different folders or do any other type of transfer. Okay, so that's about 2/3 of what I wanted to show you as far as data transfer. The next section, I want to take you inside the discovery environment, which primarily, of course, is for analyzing data, but what I'm going to show you first is how you can use it to share your data sets with your collaborators and your colleagues. So let me take you to the discovery environment. So back here on the web, I'm going to go ahead and access the CyVerse homepage. Let's do it from there. Again, this is all in the Learning Center documentation that I showed you previously. Okay, and here we are on the CyVerse homepage. It's taking a moment to load. They just launched this today, so hopefully I know where everything is, or this is the redesign. Okay, and I just clicked on the Analyze button. I want to go ahead and launch the discovery environment. And I'm going to log in with my CyVerse credentials. Okay, So this is the CyVerse discovery environment. I'm going to close this. I can actually see, and it's pretty interesting. One of the new features, or newer features, is that if you need help with something, you have this little intercom button at the bottom. So somebody's responding to a message I sent a few moments ago. I'm going to close that and answer them later. But it's a good point to point out that if you need help or you get lost, you can click on the intercom button and you can either continue an existing support conversation or you can start a new conversation to get help that you need. Okay, so I am going to go ahead and click on this data window because that's what we came here for. And on this window, if I click on my username, I can see what I've seen just a few moments ago in CyVerse or CyberDuck and also on the command line, which is the contents of my CyVerse data store. So I'm going to show you two ways to share data that are relatively quick. The first way is if you want to just share an individual file. It won't work with folders, but with individual files. And typically we would suggest this for smaller things, so certainly less than a gigabyte of data. Maybe you just want to create a link to a URL that anyone could use and they don't have to even have a CyVerse account. So I could select a file and then go up to share and then say go ahead and create a public link. And when I click on that, there is my link, and if I click on show link, I get a dialog that allows me to copy this to the clipboard. Now if I go ahead and open up a new window and paste in that link, I will get a dialog asking me to go ahead and download this. So really, in under 30 seconds, I think it's fair, I can click on any individual file that I have, and I can create a URL that I can share with anyone, and they don't even have to have a CyVerse account. They can simply download it. Now, this is a sort of, I would call, a quick and dirty way to share data. Again, very large files, you wouldn't want to do it that way because it's over the regular HTTP protocol. And sometimes those downloads can be slow or get corrupted. Also, it's not entirely private. Anyone who has that link could use it, so that may not be what you want. And we have a much more sophisticated model for publishing data. So if you want to make your data available in a publication, perhaps you want a DOI, we can do those things, but we think about using the CyVerse Data Commons for something more sophisticated than that. I won't be addressing that, but if you do have a question, you can ask as well. Now, let's say that I no longer want that link to be shared. I have that same file selected, so I can go back to the public link, and I could go here and I could actually click the delete icon that is next to this file, or rather this URL, and now that will be no longer active. If I go back to that URL and click again, I will not be able to download it. So you can use that on a temporary basis. Once you know the data has been transferred, then it's done. Fairly simple. Suppose, however, you want to share more than one file, maybe a folder, and especially, let's say it's a really large data set, so you want to make sure it gets there. Well, in this case, we have solutions, although this does require the person on the receiving end to have a CyVerse account. So I can click on an individual folder to select it, and then I can go to share, and this time I'm going to say share with collaborators. Okay, now looks like I didn't uncheck the other one, so let me go back and check and uncheck. I just want to share one thing. Oops, go back. There we go. All right, I just want to share one folder, not everything. Share with collaborators. Okay, and now at this point, I just need to type the name of the person that I'd like to share with. So I'm going to type in the name of a collaborator. I see them along with their name and institution. And then I can not only share with them, but I can decide what level of access I want them to have, read, write, or own. So back on the Learning Center, when it talks about data sharing, there are some descriptions here, and it tells me what those permissions mean. So these are sort of based on Unix permissions. And again, let's zoom out a little bit. Read permission is the lowest level, where they can essentially view things, and own, you have to be careful because that's the highest level permission, and they can do everything, including even renaming, moving, or deleting. So if you don't want that to be the case, then don't assign that level of permission. Coming back here, if I really wanted to share this, I would click Done, and then my colleague would get a notification that that data set has been transferred. And really, it is instantaneous. So as soon as I click that Done, It doesn't matter how big that data set is, they have access to it at the level of permission I've given. Let's assume that I've already shared this and I no longer want that colleague to have. I would come back here and I could click the X next to the name and also click Done, in which case their permission would be revoked and they would no longer be able to access it. Or it could even just have changed the level of permission. Anything that is shared with you appears in the Shared With Me folder. And if you click on the Shared With Me folder, you may see that there are already several other things shared with you, because there's some things that we share with everyone in this way. But you would also see the name. You'd also get, in this little bell, a notification of what's been shared with you. And again, when you get that notification, clicking on that notification will direct you completely towards the things that have been shared with you. In this case, an analysis was shared with me. If I want to see if I could check all of my notifications, I could find sometimes somebody shared maybe a folder with me that would take it to me in the data store. So yeah, somebody shared a folder with me, and then I would be able to easily navigate to the outcome of that folder and access it with the level of permissions I've been given. There are a whole bunch of other features, again, that you would kind of expect, all that I don't have time to show you here exactly. But some of those features include you can do uploads from the discovery environment. And so if you have simple files, you want to upload them that way. If you have an import from the URL, you can paste that in. You can also create new folders or create blank files. There is really a pretty fancy metadata system that we have. So if you're searching, if you do actually a smart search, you can search upon metadata attributes. Unfortunately, we don't have time to go through all of that in one presentation, but I encourage you to go back to the Learning Center and you can see more there about how to use those features. Okay, so that is it for our particular section on data sharing. In the next section, what I'm going to do is we're going to remain in the discovery environment, but now we're actually going to talk about how would you analyze data. And to do that, we're just going to give you a very simple demonstration of doing an analysis. And if you understand that, you pretty much will understand the basics of how most of the applications within the discovery environment operate. Okay, so back to the slides for a second. We've done this demo, and now we can talk about the discovery environment. Okay, so to reemphasize, it's a platform, so you can run almost any bioinformatics application that you'd normally run at the command line, except you're going to be able to run it in this easy-to-use graphical interface. It's integrated, as you saw just now, with data and also with high-performance computing. And although we won't have time to add a new application today, you do have the option of adding applications as needed. We've covered the data window, and you probably have at least a basic idea that you can see your files and folders and even do some sharing. Now we'll talk about the other two capabilities: apps, where you can select and run an application, and also analyses, where you can see the status of analyses that have been launched. Okay, so here we are again back in the discovery environment. Before I close this, let me talk about the data that we're going to use. So in this particular demo, which I failed to show the slide for, we're just going to go ahead and do a multiple sequence alignment. So we're going to take some unaligned DNA sequences in FASTA format, and we'll create a multiple alignment from that. Fairly simple, just uses one tool, muscle, and that will give us a way to sort of kick the tires, so to speak, and understand some of the basics of the discovery environment. Okay, so close the data window for, well, actually I wanted to show you the data. So the data we're going to use is in community data, and then in CyVerse training, platform, discovery environment, and muscle. Okay, so the input that we're going to be using for this here is this unaligned, or this FASTA file with unaligned DNA sequences. So most certainly text-based files within the discovery environment, you can open and preview them. Okay, so this is going to be the input for our analysis. But to actually do an analysis, we're going to click on Apps. And then from here, we can see different applications, and they appear as these sort of icons. And you can look at them by topic or by operation. So if we're looking at alignment and sequence alignment and genome alignment, etc., whatever the different categories of applications, there is some logical filtering of these. But I'm just going to go ahead in the search button and type in muscle. And at that point, I happen to know the one I want to use is this muscle 3.8.31. Yours may not look the same because I have different private workflows that allow me to see some things you may not, but this muscle is here. Immediately I can see there's a little bit of a rating system as to how many people think that this app has worked for them. If it doesn't have a rating, that's okay, but if it had a poor rating, you might want to think, is there a problem with this app, or take a look and see the notes. If I click these little three dots, which you've seen in many places before, you get additional information. So for example, the app info gives me a little bit of a description of what this application does. I can even see the last time it was used and how many analyses have been completed with it, et cetera, et cetera, et cetera. But to use the app, I can click on the name of the app and it's going to open. So I've closed the other menu, and this is what an application looks like in the discovery environment. So every application is in what we call wrapper that looks like this, and it has some options. Options that are mandatory usually have a red asterisk. In this case, I will go ahead and change this name and say this is demo, and today is 12/13/17, so I may give some comments. This is a demo. And the reason for all of this is with a graphical interface, it's hard to keep track in notes and be sort of reproducible if you're just clicking buttons. But you can leave sort of an audit trail, and in fact, we capture the information there to help you understand what apps you used, what settings you used, and we'll see that in just a few moments. My output folder is by default something called analyses, but maybe if I had a folder where everyone in my lab was using for the analyses, I might ask everyone to set that to the output. I'm not going to change that output folder now, but up in the person icon, the preferences, I can actually change my default output to be perhaps a folder where everyone in my lab has access to it. But for now, I'm just satisfied with that. So if you look, this is sort of an accordion menu. So the next arrow is sequence input data, and I have to click on browse. This is required. This will happen to browse to the last location I opened from. So I'm going to go ahead and choose that sample plants. And then I have another required thing, which is a sequence type. I'm going to select from the drop-down menu. As you saw, that was DNA. Okay, so in order to get this app running, I'm going to click on Launch Analysis. I'll get a notification that that analysis is successfully launched. And I'll see updates as that goes through the process of launching, being submitted. Now it's running, and then hopefully in a few moments it'll come back as completed. Now I can actually see the status and the history of applications by clicking on Analyses. And if I click on Analyses, I can see what is going on and a history of things I've done previously. This app is running, so I don't have access to it yet. However, I can already click on an app name, and under Analyses, I can click on View Parameters. And this is sort of the history and the details of that application that I might need and certainly want to save in a notebook of sorts so that I can do the same thing again, or at least know what I've done. Now I can see what file I used exactly. There's a direct link to it. If I click on that link, it'll open the file. And I can see the other options, like I selected DNA was my sequence type. I can click Save As, and this would allow me to save this as a plain text file in a place of my choosing. This app is not all that sophisticated. If I go to an app that had more settings, so let me try to find something that is more complicated. I was looking for the last time I might have used something like Top Hat. I could also search for it, but I was lazy. Same thing here. If I click on View Parameters, I can see all of those parameters. So again, I shouldn't be guessing at what I did the last time I ran an application. The other thing that's cool here is if I take an analysis and say relaunch, that application will relaunch using the same exact parameters as previous. And this is important because rather than clicking 20 different settings here, I have everything that I used last time. In fact, if I want to make a tweak, now rather than tweaking 10 things that I tweaked, I can just say, oh, I forgot I want to make the anchor length 7 this time. Okay, so the relaunch button is useful. Back to analyses, okay, I saw that my application has completed, but in fact, it doesn't say that here. I saw the notification, so you may have to click refresh, and it does say that it's completed. Now, if I want to actually view the results, I can click on the name of that application, and it takes me directly to the output, in which case if I clicked on one of those, I can actually see now I have a line DNA sequence. Okay. So, of course, every application that you might use, whether you're doing a genome alignment or whether you're doing anything else in the discovery environment, maybe several more steps or several more applications. However, the sort of modus operandi is the same. You go into data, you organize the data that you've uploaded. Maybe you've already set up a way for common sharing. You would then select the application or applications that you need. There's also options, though they didn't show you here, to use a workflow. And then once those applications are running, you can certainly check on the analyses, view the history of different analyses. You can even share an analysis with someone else who's a CyVerse user. And there you have it. It's really a web-based interface. Once I click launch on an application, that will run until completion. I can close my computer. It doesn't matter if I'm logged in or not or connected to the internet. I can just run it and it will go ahead and run. So that's it for just giving you a very basic introduction into the discovery environment. I will remind you again, if you have questions, please at the bottom of the screen at any time, let me know about those. But I'm going to move on to our last demo, which is using cloud computing in the Atmosphere environment. So let me return to the slides. Okay, so we don't need this anymore. And here we are on the slides. Okay. So Atmosphere. Atmosphere, as I stated before, is giving you access to hundreds of virtual machine images. But if you've never used cloud computing, you may be wondering what that means. So I'm going to give you some definitions. It is very flexible because it's fully gives the ability to customize how you install your software, use your software. And again, it's integrated with our computing and data resources. So first, let me talk about what is cloud computing. And in fact, to do that, I'm going to define two key words for you. Those words or terms or concepts are image and instance. So those are the two things we need to figure out. What is an image and what is an instance? Well, first, when we talk about image, I'm going to use a little bit of a scenario here. Suppose you have an old computer and you buy a new computer. You want to take data and you want to take whatever images, whatever you have on your old computer and put it onto the one that you just bought. So you might take a hard drive, an external hard drive, and copy documents, files, whatever, onto the hard drive. But there is a different type of file that you could make, which is called an image. And an image is not an ordinary file. It's actually a complete backup, a complete mirror of your disk. It's sort of what you use on your computer to actually restore your computer if it's damaged. It contains the operating system. It contains individual softwares that you've installed and the configuration of those software, and it also contains a copy of any data files. So whereas documents taken from a hard drive are still those documents or individual files or individual images, an image file can actually completely clone the old machine onto the new machine, including everything down to the image that you have on your desktop of your cat. The software that you've installed, you no longer need to reinstall that software. It's all there. So an image is really a complete system. The next term is something called instance. So in the CyVerse cloud, we have physical resources, hard disks, memory, CPUs, and when you come to us for a portion of that, an allocation is what we call it, you're asking for a certain amount. So we give you that certain amount, and then to actually make that into a functioning machine, you also need an operating system. But when you put the two together, the thing that you have now is called an instance or virtual machine. So this is your own private computer that existed only 10 or 15 minutes after you requested it. That's the first time it came into existence, so to speak. And it's accessible to you over the web or through an IP address. So I'm going to show this, but first, what are some of the advantages? Well, first, you can choose an existing image, which you can customize. We provide for up to 16 core 128 gig machines, which is pretty powerful. It may not be something you have lying around, so it's nice to know when you come to CyVerse, in 15 minutes you can have access to something like that. You can access via the shell, or if it has a desktop, through a virtual network client, VNC. And you can also share your images with other users or make them entirely public. There are a lot of reasons to use this. Here are just a few. So if you have software right now that monopolize your laptop or your desktop, you could run them on Atmosphere. If you have programs that require their own graphical interface, we couldn't integrate those in the discovery environment, but you could use them on Atmosphere. With Atmosphere, you have full administrative or sudo access, so you can install things without asking an administrator to do it. If you are worried about reproducibility, certainly in teaching, I am. I want every student to be working on exactly the same machine, not 20 students with 20 different laptops of their own that I might have to figure out how to get things installed, or they didn't install things, or whatever the case is. And also, people release software in this form where they've installed things and data sets, and they actually use it as a training or a dissemination mechanism for their software. So in this demo, I'm going to show you how to connect to an Atmosphere instance. You could use any of these services, including a VNC Viewer, which is a free software, as well PuTTY for Windows if you don't have a Bash terminal, and Mac and Linux already have a terminal. Okay, so let's go and take a look. I am going to go again to the CyVerse homepage, and I'll remind you that if you are at the CyVerse Learning Center, let's actually go to Learning Center Home. All of this is there in the Atmosphere guide, which is the link in the Adobe Connect room. Everything is there for you, so you don't have to get too lost, but if you have questions, please let me know. So from here, CyVerse homepage, we are going to go ahead to Analyze. And we're going to go ahead and launch Atmosphere. Now, as it turns out, I am already signed into Atmosphere because I'm signed into the Discovery environment. And alarmingly, I think, well, first let me click on Login. Okay, I'm presented with a dashboard of my status, and alarmingly, I'm over my allocation. So you're granted an allocation every month, and that refreshes every month. But if you need more allocation, you can get that. Allocation is simply the amount of time that you can use a given computer or given virtual machine of a given size. So what I'm going to do is just sign out and go into a test account where I've already set something up, so that I can use something else that I've already set up for myself. Okay, so thankfully this one has not used their allocation. You can see a history of the virtual machines that you've used. If I click on Launch New Instance, what will happen is it'll take me to a collection of images because I need to choose an image to start with. If I search for topic, for example, RNA-seq, I might find that people have already built images where there are a bunch of tools installed that they like to use. If I were to click on any one of those, then I could click on Launch, and then I can decide what size I'd like to use. A little caveat is that not every size is available at every given moment, so there are some limitations. Another caveat is that when you come to use Atmosphere for the first time with your account, you actually have to request access, and so to do that, we'd encourage you to go back to the CyVerse user home portal, and from there, this is the place where you signed up, where you clicked on Login to CyVerse. You can request access. This is described in the CyVerse guide. I'm not going to do this right now, but I'll just mention that to you. And if I was really going to launch this, which I'm not, I would click Launch Instance. And then within a few minutes, I would have the machine available to me. I would also get an email letting me know. Usually it takes 10 to 15 minutes. Now, if I go back to my projects, I already see that I have a project, and I already have an instance that's there that I launched maybe an hour ago. So I'm going to go click on that, and I get some details about it. So this is my instance. Here's an IP address that I can access it, but I can already from here access it. So if I click on Web Shell in a new tab, I will be given a command line instance. And this, unfortunately, I cannot make bigger, I think. Let's do it this way. Yeah, this one doesn't enlarge, so sorry. But I do have access to this computer, so if you're used to the command line, you can see that I have access there. Any Linux command that I'd use is associated with my CyVerse identity. Okay, I'm going to close this. This happens to have a desktop, so if I click on Open Web Desktop, again through the internet, I could see that I'm connected to this virtual machine. I can then go ahead and see what's installed. In this case, in applications, I could go on the internet, which is not hyper useful, but I could launch any particular application that's there. If I had things installed, I could use that. Okay. Of course, I could do any of this from my computer as well. So if I copy this IP address, the same thing on a terminal, I could SSH in. So in this case, it would be SSH that identity, or your CyVerse identity at that IP address. Yes to connect my CyVerse credentials, and then I would have access to that. Or if I had VNC on my local machine, same thing. Connect, yes, that identity. I think I messed that up. And again, here it is. It's the same machine. If I make a new folder on my desktop, okay, that folder will appear on the desktop there. Okay, so the point of this particular system is essentially that from a list of existing images, you can, at will, choose a software setup that has things pre-installed, or one that we call a base image that doesn't have very much installed, it's sort of bare bones. You can customize it. You can then, within a few minutes, use it. And it's probably, in many cases, a much more powerful machine than you'd find in your laptop, maybe even in your lab. You can install your own software. You can use it at will. And then when you are done using it, you sort of return it to the ether, which I'm going to show you in a moment by terminating that instance. So let's go back to the Atmosphere interface, and I'll show you what to do once you're finished. So back on the Atmosphere interface, there's a couple things you need to know. Right now, this instance is listed as active, so I have a green dot here. Let me make that a little bit bigger so you can see it, and we'll switch to my computer screen. There we go. So this is active. It's worth mentioning that when my Atmosphere instance is connected for the first time, I don't actually have access to my CyVerse data store. So I need to transfer data from my data store into my newly created instance. I could use iCommands to do that. And there are also some options to transfer data in other ways. You could also create a volume within Atmosphere, and all of these things are described in the Atmosphere guide. Once I'm done, I have a couple of options. If I've installed software and I want to come back to this and use this on a frequent basis, I might decide to image this, which would mean making a new image where I've installed those software, and next time I wouldn't have to install them. If I know I'm going to come back to this maybe in a month, then I might do something like shelve. So I can save this particular configuration and everything that I've done, and that would be the shelve. That will save the instance state, although I'll get a new IP address the next time around. There are similar ones like suspend and stop, and these are described again in the documentation, but you have the option to do that. When you are really done and you don't intend to use this again, the final step is to click on delete. The delete the instance will completely erase any data and anything associated with that. So do not do this unless you are sure you are not going to use that again, because any data is going to be lost. So you'd want to transfer that data off to another machine or another location before you would do that. In this case, I'm not going to use this anymore today, so I'm going to go ahead and say yes to deleting that instance. It would delete, and I would not have access to it. I'd have to launch a new one. So with that said, I'm going to take you back to the CyVerse homepage, because that's the end of our demos, and we're at the end of our time here. And just want to point out a couple of things. Of course, this is new, so I hope I have them all right. Back at the CyVerse homepage, what I encourage you to do is check out the knowledge base, where you can see some links to tutorials and other learning resources. And I also want to show you where you can look at upcoming events and workshops. So those are important if you want learning opportunities, and we in fact also do workshops and can come in many cases to your location to do workshops. And this is a place really to connect with us and ask more questions. So with that said, I'm just going to allow a few more seconds for anyone who may have some questions. We really appreciate all of our users and appreciate you giving us your time. If I don't see any questions, I'm going to go ahead and end the webinar, and I will push you to a very short survey that we can get a little bit of feedback from you. We really do encourage you to make the best use of your CyVerse account, make the best use of us in terms of asking questions and getting help from us about anything that you need to do with CyVerse. And we look forward to meeting you even in person in some cases as we do workshops or maybe see you at some scientific conferences and other events.